We're going to continue our discussion of biochemistry today and pick up where we left off last time. Last time we talked about protein shape, and now we need to move into protein function. And of course, protein function is intimately related to the shape of the protein. So one of the functions of at least some proteins is that some proteins are enzymes. And an enzyme is described as a protein catalyst. Chemically, a catalyst is a substance that speeds up a chemical reaction without being consumed in that reaction. So those are two really important concepts to understand about enzymes. So they can speed up reactions tremendously. The typical speed of an enzymatically mediated reaction is about 30,000 reactions per second, 30,000 cycles per second. And then again, the other aspect of it is that they can be reused over and over and over again. So a lot of the reactions that are required for us to be alive would not spontaneously occur at a rate fast enough to maintain life. So enzymes are critical for life. Now, they do this by lowering what's known as the energy of activation of a reaction. And what this is trying to show us is energy related to reactions. And most reactions require an initial input of energy before they will run on their own. Basically, they'll spontaneously continue after a certain point. And so what enzymes do is that they lower the energy required to get a reaction to spontaneously occur. And because of that then, there's less energy required per reaction, and so a certain unit of energy will conduct more reactions. All right, so let's look at some of the things that enzymes do. So some enzymes we describe as being catabolic, which means that they will take large structures or large molecules and break them into smaller molecules. Anabolic enzymes will take small molecules and build larger molecules from those. And then also they tend to rearrange molecules. So using the overhead camera in our Legos again, we can go ahead and look at what I mean by this. So again, a catabolic enzyme would bind a large molecule and break it into smaller molecules. An anabolic enzyme would bind smaller molecules, combine them to form larger molecules. And then there's always the potential to rearrange a molecule as well. Now, these aren't the only things that enzymes can do, but these are three pretty easy ones, I think, to recognize and understand. Additionally, enzymes are very specific in what they do. A single enzyme does not carry out lots of different types of reactions. It carries out one specific reaction. So if you do have, and so typically then what happens is you'll have another number of enzymes working together to accomplish some large task. Now, one of the models for how enzymes function is the lock and key model. And in the lock and key model, what we have is an enzyme, and you'll notice that there are certain shapes here, and these are known as active sites of the enzyme. And the enzyme then will bind a specific substrate that has a complementary shape to the active site. So the shape of the active site is critical because it will bind only a particular substrate. And this is why enzymes are very specific in what they do. So we start with the enzyme and substrate. Now, in collisions through the cell, they potentially bind together, forming an enzyme substrate complex. And so this puts them in a particular position for either bonding, or in this case, it's going to break the bond. So stress may be put on that bond, and that will cause the bond to break. And then we end up with our products here. So we end up with enzymes and product. But the key thing to understand then is that the shape of the active site goes with the shape of the substrate. And at the end of the process, we end up with an enzyme that can be reused again. And it will continue to carry out the reaction as long as substrate is present. So a little animation to show us this as well. Again, we're seeing the active site. And so it will bind a substrate that's complementary, act on it, release the products, and the enzyme can be reused again. Now, another model for how some enzymes work is what's known as the induced fit model. And so again, an enzyme would have a particular shape, and it would have active sites where it would bind substrates. But the idea is that when the enzyme, and I'm going to be the model of the enzyme this time, and so I would bind one substrate, and in this case another substrate. And when I bind the substrates in, that would actually change the relationships between side chains and so on, actually change my shape so that I put them together so that they can bond together. Then once they bond together, that changes, again, covalent bonds and polarity of my molecule. I'll release it and go back to my original shape. So the induced fit model then, after binding of the substrates, it changes the shape of the enzyme to put them in ideal position to bond, then releases them and goes back to its original shape. So the general idea is the same in that the enzyme starts and ends in a particular shape and can be reused over and over again. Now, not all proteins are enzymes. Some proteins are what we call transport molecules. And there are two types of transport: passive transport, which does not require any kind of cellular energy. It's called ATP, and we'll talk more about that later. And an example here is that we have our transport protein, and some of these are just literally channel proteins and allow materials to pass through a membrane. So what we're looking at here is the outside of the cell, the inside of the cell, cell membrane, and here's a transport protein embedded in that cell membrane. Oops. And so what it's going to do is allow particles to pass through the membrane. So it allows particles to transport through the membrane. So in passive transport, there is no utilization of cell energy, and particles can only go from where they're more concentrated to where they're less concentrated. Now, in active transport, again, cellular energy is required for this. And the other aspect of this is that particles can go from where they are less concentrated to where they're more concentrated. So in this animation, we're going to see sodium's being transported in one direction, potassium's being transported in the other direction. And you'll notice again that there are shapes, complementary shapes. Here, energy is added. And then the binding sites here will be complementary to the potassiums. And then they can be transported across the membrane. And we'll be talking more about transport proteins later in the course. Still, other proteins can serve as receptor proteins. And so receptor proteins are proteins that we find in the cell membrane, and they will bind other proteins or other structures. So again, they have complementary shapes. The receptor protein binds some molecule, and that triggers a change inside of the cell. So any kind of a protein that binds some substance out from the outside of the cell is known as a receptor protein. Still, other proteins may serve as carrier proteins. And I'm going to go back to this same animation, and again, this would be our receptor protein. But I want you to look at the protein then that binds to this, and I'm going to have you use your imagination just a little bit. Again, notice that they have complementary shapes, but what a carrier protein does is one that potentially binds to a receptor. And carrier proteins are attached to other substances. So one of the examples of this is that when we absorb fats, we need to deliver them to the liver for processing. And so we'll attach proteins to those fats. And those serve as carriers and make sure that they're delivered to the liver. So if we imagine that this little triangular piece right here was actually the carrier protein, and the rest of this was a fatty substance, then what happens is that the carrier molecule binds to a complementary receptor protein and makes sure that that substance that's attached to then gets delivered to the appropriate cell type. So again, this little part here would actually be the carrier protein. The rest of this might be some other substance. So carrier proteins then just make sure that substances get transported to the appropriate cells in our body by binding to specific membrane receptor proteins. Proteins also play a role in communication between cells, different parts of the body, and they can do this through hormones and neurotransmitters, which is how nerve cells communicate with one another. And there are other types of communicating hormones and chemicals as well. So again, we would still rely on a receptor protein. It's just that now either a neurotransmitter or a hormone would bind to that receptor, triggering some kind of change inside of the cell. So a hormone then can be secreted in one part of the body and have an effect on another part of the body. And this is also how nerve impulses get from one nerve to another nerve. And then a lot of proteins are connective. They connect cells to cells, tissues to tissues, bone to bone, muscle to bone. All of these kinds of things would be examples of connective proteins. And so in the extracellular matrix outside of our cells, there are a number of different connective proteins that we find there, again, connecting things together. Collagen is an abundant protein in our body, and it's an important connective protein, for example. Also, proteins play an important role in the immune system. Our antibodies, for example, are very large, complex proteins. So again, proteins have just a tremendous number of functions. Another function is that, and we'll talk about this later in the course as well, is that protein, just by their presence, soluble proteins that are dissolved in our body fluids, help regulate where fluids are found in our body. For example, you may have seen pictures of starving children in particular that will have swollen abdomens. They're protein deficient, and so they cannot hold fluid in their bloodstream, so it leaches out of their bloodstream. So proteins are responsible in regulating fluids and keeping fluids where they're supposed to be, in this case, within the bloodstream. Now, another major class of organic molecule, biomolecule, would be lipids, which are also referred to as hydrocarbons. So lipid is a descriptive term for anything that is kind of greasy or oily in texture. The term hydrocarbon is a chemical term that's telling us that they're composed of hydrogen and carbon. So they're composed primarily of carbon-hydrogen bonds, greasy, oily in texture, which is also just another way of saying they're nonpolar, which also means that they are not water-soluble. So lipids have a number of functions, primarily as an energy source. They are also structural in some cases. We'll talk about some structural lipids. They can also play a role in communication between cells, different parts of the body. And then also they can play a role in immune responses. Sometimes lipids are part of what our immune system recognizes as either being foreign or native to our body. And they have other functions as well. But again, these would be the primary functions of lipids. Now, again, there are a number of types of lipids, and we're going to start off talking about fatty acids. So a lot of lipids are composed, at least in part, of fatty acids. Now, there are three types of fatty acids. Actually, there are three major types that we're going to talk about. The first being saturated fatty acids. Saturated fatty acids are composed of long chains of carbon. So they can be 20, 30 or more carbons in length. And all of the carbon bonds, except for the carbon-carbon bonds, but any extraneous bonds to the carbons are quote-unquote saturated with hydrogens. So they bond hydrogens all around. So we have a methyl group on the end and then hydrogen, hydrogen, hydrogen. So we have single bonds between carbons and then all available bonds quote-unquote saturated with hydrogen. And then at the terminal end, we will find a carboxyl group, COOH, which is an organic acid. So this carbon-hydrogen combination is nonpolar, which means that it really does not dissolve in water very well. And so that's why they're fatty, also fatty in texture. And also, the acid group gives it an acid characteristic. So it's a fatty acid chain. So the saturated fatty acids then have certain characteristics. We have single bonds between the carbons, as we just saw. They tend to be more solid at room temperature. And when they're transported in the bloodstream, they tend to get transported as low-density lipoproteins. So as I mentioned earlier, when we absorb fats and fatty acids in the bloodstream, they need to be delivered to different parts of the body, so we attach proteins to them. So these low-density lipoproteins are called LDLs. They tend to stick to vessel walls, especially if there's any damage to a blood vessel wall. This then leads to plaque formation and contributes to cardiovascular disease. So saturated fatty acids are important. They're really important for energy. They're used in a variety, in the construction of other lipids. But one of the downsides to saturated fatty acids, again, is that they're transported as LDLs in the bloodstream. That contributes to plaque and cardiovascular disease. Now, another class of lipid would be unsaturated fatty acids. Now, unsaturated fatty acids are of two types, monounsaturated, which means that they have a single double bond between carbons, And then there are also polyunsaturated fatty acids which have more than one double bond between carbons. So what that means then is that, again, this is a monounsaturated fatty acid. If we look at the carbons, there's a single double bond here. So that's an example of an unsaturated fatty acid. And again, these can be extremely long chains again. And again, we'll have our terminal acid group. So long chains of carbon-hydrogen with at least one double bond is an unsaturated fatty acid. So at least one double bond between carbons. They tend to be oils at room temperature, and so again, just that tiny change changes characteristics of the fatty acids. So instead of solid at room temperature, they tend to be oils, and that's how you can recognize that something is higher in unsaturated fatty acids than saturated fatty acids, by looking at whether it's an oil or solid at room temperature. Also, that tiny difference means that when they're transported in the bloodstream, they can be more compacted. They are transported as high-density lipoproteins, HDLs. And HDLs are less likely to stick to vessel walls, less likely to contribute to plaque formation, and less likely then to contribute to cardiovascular disease. So if you have a choice in your diet as to whether to consume saturated versus unsaturated fatty acids, you definitely would want to choose unsaturated fatty acids. Now, some fatty acids are considered essential fatty acids. And essential fatty acids, we've heard that term before with amino acids. If something's essential, it means it cannot be synthesized by the body. So it means we have to ingest our essential fatty acids. And this includes omega-3 fatty acids, which you may have read about. And they tend to be high in seed oils, such as flaxseed, and also fish oils. And a couple of examples are linolenic acid and linoleic acid, which we find, again, primarily in oils, seed oils. Now, in addition to the essential fatty acids, which are unsaturated fatty acids, I should add, there are also partially hydrogenated fatty acids, also known as trans fatty acids. So what happens is there's a process known as hydrogenation, where fatty acids are subjected to an increase in temperature and high pressure. And unsaturated fatty acids can be converted to saturated fatty acids in that process of hydrogenation. But some only go through a partial hydrogenation, and as you can see, what happens is that it changes the orientation of the hydrogens. So chemically, they have the same formula, but structurally, they're slightly different. And it's, again, a tiny, tiny change, but it has important consequences. Now, the cis chain is the naturally occurring chain. And actually, if I can go back for just one. So the cis chain is a naturally occurring chain. The trans chain then is an artifact of this hydrogenation process where fatty acids are partially hydrogenated. So cis chains typically have kind of a kink in them like this, and so these fatty acids will have a bend in them. Whereas once they're hydrogenated, they look and behave more like a saturated fatty acid. And so that's the problem with trans fatty acids. They get a lot of, you hear about them in the news a lot, but they essentially, even though they're still unsaturated, they behave as if they're saturated. And by that I mean they'll tend to be more solid at room temperature than a cis fatty acid would, which is, again, the cis shape is the normal chain. And as a result, then they tend to be transported in the bloodstream as low-density lipoproteins, and of course that contributes to cardiovascular disease. Now, additionally, trans fatty acids have fewer biological uses because, as I mentioned before, fatty acids can be used for a lot of different things in the body, potentially. And we're going to talk about some of the other kind of lipids that can be built from fatty acids. But trans fatty acids have limited biological uses. And so a higher percentage of the trans fatty acids will go to fat storage than would cis fatty acids or just naturally occurring saturated fatty acids. So that's an important consideration as well. So if you are in a diet where you're getting lots of packaged foods, or if you're eating in a lot of fast food type restaurants where trans fatty acids are used, if your diet is high in trans fatty acids and saturated fatty acids, then it makes it more likely that you will store those fatty acids as fat and also contributes to cardiovascular disease. So you really want to try to eliminate the trans fatty acids and then substitute unsaturated unsaturated fatty acids for saturated fatty acids when you can. Okay, now another category of lipids are fats. And fats are called triglycerides, meaning they're composed of one molecule of glycerol and 3 fatty acids. And those fatty acids can be saturated or unsaturated or trans fatty acids. So, same old story, glycerol, fatty acids, we're going to put these together. So we're talking about a dehydration synthesis to put these together. And that's how we get from glycerol and fatty acids to a triglyceride, also known as a fat molecule. Another look at the same structure. So again, almost any kind of fatty acid can be used to make fats. And then the triglycerides are how we store excess lipids. So we store those in our adipose tissue, and we save those for a rainy day because they have a lot of energy. Still, another category of lipids are phospholipids. Phospholipids are a structural lipid or structural hydrocarbon. And again, you don't need to worry too much about the structure here, but they have what we call a hydrophilic or water-soluble head, and then typically 2 fatty acid tails. And again, these can be saturated or cis fatty acids, unsaturated fatty acids in the tail here. So we refer to these as the tail. This would be hydrophobic or not water-soluble, and then a water-soluble or hydrophilic head. So we normally just represent them this way with a circle for the hydrophilic head and then 2 lines to represent the hydrophobic tails, the fatty acid tails. And what's important about phospholipids is that they will spontaneously arrange themselves with the tails towards one another, and they are a primary component of our membranes, cellular membranes. So we'll be talking about the structure of the cell membrane and plasma membranes within our cells. So the plasma membrane structure is primarily a biphospholipid layer. A bilipid layer is the typical structure of all plasma membranes in all organisms, everything from bacteria to human beings. And then that creates a barrier, again, between potentially the inside and outside of a cell, or the outside of an organelle and the inside of some type of organelle. Cholesterol is another example of a lipid. Cholesterol is a complex series of rings. Cholesterol is also found in cellular membranes, and so it's important there. And then also it can be converted into other substances such as steroid hormones. So testosterone, estrogen, and their relatives are all derived from cholesterol and are also lipids. Waxes would be another example of lipids. I'm not really going to show you the structure of those, but those again would be another category. And then there's some molecules that we find in our bloodstream called prostaglandins that are related to control of temperature. They actually have a lot of metabolic functions in our body, which would be another example of lipids. So there are other functions for lipids, but these are all very important and primary functions for lipids. And examples of lipids. Now, all of the major molecules we've talked about to this point, carbohydrates, proteins, and lipids, all have energy value to the body. And so when we talk about energy, the primary unit of heat energy is the calorie. And 1 calorie is the energy required to raise 1 gram of water 1 degree Celsius in temperature. And unfortunately, on our food packaging, we have what's known as a nutritionist calorie. And the nutritionist calorie is equivalent to 1 kilocalorie. So a kilocalorie is the heat energy required to raise a kilogram of water 1 degree Celsius in temperature. So when you see the word calorie on any food packaging, it would be what a chemist refers to as a kilocalorie. But the bottom line is that it is a unit of heat energy. So again, kilocalorie is a chemist. Calorie on packaging is a chemist kilocalorie. So when you look at foodstuffs, then food has different caloric value. For example, a carbohydrate has approximately 4 kilocalories per gram. Proteins also have approximately 4 kilocalories per gram. But lipids, on the other hand, because of all of the length of the carbons and so on, have about 9 kilocalories per gram. So again, that's really why we are adapted, to like fatty substances so much. It has tremendous energy value, and we can get a lot more, energy value for each gram of material that we consume that way. So when you look at the United States, for example, then, we don't really have a whole lot of nutritional problems related to deficiency. Most of our nutritional problems are related to excesses. So this is one of the things to keep in mind then, that, most people have a trouble ingesting too many calories. So the fatty materials have more calories, and that's why they're problematic for us. Also, if you like to party, ethyl alcohol has about 7 kilocalories per gram, so which is almost as much as other lipids. So that's also something to keep in mind. So the other thing I want to come back to then, in kind of summary to the macromolecules we've talked about then, is that in almost every case, we've talked about basic monomer units then that can be assembled into polymers through dehydration synthesis. And then, of course, the other side of that is that we can take those molecules and break them back down into monomers through hydrolysis reactions. So one of the things I want you to understand, and we'll be talking about this more in other modules, is that when we look at nature then, there's a recycling of materials. And if we talk about the basic monomers, the different monosaccharides, amino acids, the lipids we've talked about, they're not unique to a particular organism. For example, there's not human amino acids and mouse amino acids and banana amino acids. Amino acids are just amino acids. What determines whether we're a human or a banana or a mouse is essentially how those amino acids are assembled to form proteins. So again, the basic concept then, if I can go back over to the overhead view, is that what happens then when we ingest food materials, and let's use proteins as an example, is that through hydrolysis reactions we will go ahead and separate these and break these down into individual amino acids through hydrolysis reactions, and that's what happens in our intestine. And I won't have time to reassemble all of this. But then what happens is that these individual amino acids are taken into our cells, taken into our body. And then what we do is take exactly the same amino acids and we reassemble them into a completely different order and a completely different type of protein. So what makes us human isn't the amino acids that we're ingesting. What makes us human is that we will build those amino acids into a protein that is a human protein. And that's ultimately determined by our genetic material. So the molecules of nature then recycle in nature through the food chains. And then we'll be talking about how energy has to flow through ecosystems to make this all work.